Okay, so that's my goal of this talk today, is to introduce you to the first experiment that we've done and the rationale as to why we use this particular spatial platform to address this particular question that we're interested in addressing in looking at lineage differentiation in the embryo. And hopefully this can begin to stimulate discussion around the different spatial technologies that are starting to emerge right now that we as biologists are starting to interact with. So that we can start to think about which technology best suits the question that we're trying to ask. And I think that's a really important concept as these new technologies roll out. So I wanted to begin with this slide here. So in our lab, we are stem cell and developmental biologists, and we're very interested in engineering models of reproductive organs. So this is an engineered model of the ovary. It's a mouse ovary. And the way we achieved this is we used induced pluripotent stem cells to differentiate the early germline, which in this particular model organ is in blue. So the induced pluripotent stem cells have a reporter, which is a cyan fluorescent protein. And this is driven by a specific regulatory elements that are expressed only in oocytes, and in particular, this signal gets brighter and brighter as the oocytes grow. So these oocytes are iPS cell derived within this model organ of the ovary, and the somatic compartments are derived from embryonic progenitor cells in a single cell suspension that we combine together, leading to the self-assembly and creation of this model of the organ over 3 weeks. We're good. We're good. Sorry. Okay, great. So this model of an organ is not an organ. So when investigators talk about organoids using organoids model, the general public hears this as you're making an organ in a dish. And that's not what we're doing. And actually, that's not what any of us in stem cell biology are doing, is making organs in a dish. We're making models of aspects of organ development in a dish. In this particular model, the model of the ovary that we're making, we're modeling 3 major cell types and the interactions that occur between those cell types. So in this model of the ovary, we're making the germline in blue from iPS cells, and we're modeling its interactions between the germline and the ovarian somatic cells, which are the granulosa cells. This is a very specific ovarian epithelia that signals to the ovary to instruct the, signals to the oocyte to instruct it to grow and enter meiosis at the appropriate time. In this model, we have a 3rd cell type that's not stained here and you can't see, which is the stromal compartment of the ovary. And the stroma contains the progenitors for generating the products that are necessary to create the hormones so that this can become an endocrine organ. And so what we're able to show in the lab is that these models of the ovary, although they don't have blood vessels, they don't have nerves, does have the capacity to create endocrine activity and is able to produce estrogen, progesterone, and testosterone, which are the major endocrine products of the ovary. And it does this entirely in vitro. So these models that we make as stem cell biologists in the lab are fantastic for understanding aspects of organ development and organ function, but it's not an organ itself. The way stem cell biologists work with induced pluripotent stem cells is to take lessons from developmental biology. So we're building organs in the lab, me building ovaries, Michael Hicks, who's building muscle cells. What we need to do is take lessons from human embryo development. So the ovary and many other reproductive organs develops from a mesoderm called the intermediate mesoderm. So this is a cross-section through a human embryo in diagram that you can see. And it's essentially highlighting the 3 major mesodermal cell types that are formed in the first 2 weeks post-fertilization. They are the paraxial mesoderm, which will go on ultimately to contribute to somite formation, and the lateral plate mesoderm, which will ultimately contribute to the mesoderm of limbs and other parts of the lateral elements of the body. And this tiny little bit of mesoderm in here that you may never have heard of before is the intermediate mesoderm. And that intermediate mesoderm is fated in order to form reproductive organs, the ovary and the testis, as well as the reproductive tracts. The intermediate mesoderm is also fated to form the kidneys, as well as the adrenal gland. So as a scientist, as we're generating these cells in the lab, we need to think about what are the molecular processes involved in intermediate mesoderm formation. And then in our particular case, what are the molecular processes that drive intermediate mesoderm specifically to the formation of the ovary or the testis and not the adrenal gland and the kidney. So these are the sorts of decisions we need to be able to go through in the lab as we're building these models. So this is looking at the human embryo at 3 weeks, again in diagram. So I wanted to talk about some more cell types that you're going to hear me discuss today. So one cell type I haven't mentioned yet is the germline. And so the germline does not form in the ovary. So if we're generating an organoid model or a symboloid model of an organ, we need to have the key cell types. Often these cell types are originating from a similar progenitor, the intermediate mesoderm in this case. However, the germline forms much earlier in human embryo development and is set aside at around the time of implantation as the amnion is being specified from the epiblast. So what this means is we need to make germline separately first before we combine it together with the gonadal-somatic cells to create these organoids. And so this is the germline that migrates into the embryo and ultimately into the genital ridges through this mesonephros structure, which is a primitive kidney that degrades in the second trimester, but it's very important in the first trimester of human embryo development. And the genital ridges, which will ultimately form the ovary and testis, are forming on the medial side of these mesonephric genital ridges. So how does the ovary and the testis form? So ovary and testis formation begins with the formation of a structure called the bipotential gonad. So this bipotential gonad is first developing in the 4th to 5th week post-fertilization. So I hope what you're appreciating here is that when we're looking at these early events in human embryo development, it's very difficult to study them using human embryos because we're looking at timing of human development when a woman may not even realize she's pregnant. And of course, understanding human embryo development requires access to human fetal tissue. So therefore, a lot of lessons about early human embryo development translating knowledge from other species like the mouse to human. So this is knowledge in the mouse that I'm telling you about now. So here's our mesonephros, these large embryonic kidneys that are found in the first trimester embryo. These are formed before the permanent kidney forms, which is called the metanephros. So the mesonephros is the second kidney. And the mesonephros has ducts, the Müllerian duct and the Wolffian duct. And ultimately, those ducts will give rise to the reproductive tracts that are found in males and females. And when I'm referring to male and female at the current time, I'm talking about biological sex and not gender. So XX and XY. Based on knowledge in the mouse, what we know is that the surface epithelium of the mesonephros in the posterior end of the embryo will begin to express the transcription factor GATA4, and that GATA4 positive epithelium is essentially the beginning of bipotential gonad formation. This epithelium that's GATA4 positive will start to proliferate to form a nascent bipotential structure on the edge of the mesonephros, and these germ cells that were specified at around week 2 of human embryo development begin to migrate into this mesonephros, and the basement membrane will start to break down between the mesonephros and the developing ovary and testis, the bipotential gonad, and the germ cells will start to intermingle in this structure. This bipotential gonad has absolutely no evidence of sex determination, so there's no sex-specific cell types that have been identified at this point. So how does sex determination occur? So what drives the formation of that bipotential gonad to form either a testis or an ovary? So animals have developed a lot of diverse strategies to be able to form either a testis or an ovary. And I just want to show you 2 examples here. And this is from a paper of my colleague Humphrey Yao that was published in 2005. Reptiles use temperature-sensitive mechanisms to drive the formation of the ovary and testis, and therefore the formation of male or female animals. And so this is actually really important because as we're thinking about climate change and the species that exist on Earth today, we have a number of species that are using temperature-sensitive aspects of sex determination, leading to sex biases in some of the species on Earth today because of their temperature-sensitive sex determination. So in reptiles, estrogen and in a temperature-sensitive environment will drive formation of the ovary, high estrogen, and no aromatase, but low estrogen will drive formation of the testis. Thankfully, eutherian mammals, of which we are one, animals with placentas, do not use temperature-sensitive sex determination. We use a genetics-driven method of sex determination. So in eutherian mammals, what we appreciate is that there is a key gene called SRY, which is which is found and expressed from the Y chromosome of XY individuals. This gene drives formation of the testis. There is no equivalent of SRY in females. Instead, the environment for forming an ovary is driven by a high Wnt environment, in particular Wnt4 and arspondin. And so there's these competing pathways for ovary and testis formation in the body. And so again, when we're making these cell types in the lab, we have to think about these competing pathways in order to generate an ovary or a testis from the bipotential gonad. So sex determination is the conversion of that bipotential gonad to either an ovary or a testis. And this occurs at 6 to 7 weeks post-fertilization. So we're really early still in the first trimester of development. And this is actually when we can begin to start to pick up human fetal tissues to be able to understand the cellular molecular events involved in testis and ovary formation. And so the bipotential gonad essentially starts to receive signals from SRY, which leads to the expression of the transcription factor SOX9, and that drives testis fate. And the testis fate is essentially the creation of these epithelial tubules called seminiferous tubules that contain inside them the SOX9 positive Sertoli cells that instruct the germline cells to adopt a male germline fate that forms prospermatogonia. In this high Wnt4 environment, which occurs in the absence of SRY and SOX9, we begin to have the expression of another transcription factor called FOXL2, and FOXL2 drives the expression of those same progenitors that form Sertoli cells in the testis to form granulosa cells in the ovary. So FOXL2 drives granulosa cell formation. And that is completed by 7 weeks post-fertilization. So the United Nations predicts that around 140 million babies will be born this year around the world. And for those parents who are going into their obstetrics appointments, many of them will be asked in the second trimester, do you want to know the sex of your baby? And most parents, I did at least, will say yes. And so knowing the sex of the baby, which is seen at ultrasound in the second trimester, is driven by all of those events I just talked about in the first 4 to 6 weeks of development, the formation of the bipotential gonad, sex determination to create the immature ovary and testis. The testis will begin to start producing testosterone, which will start to lead to external sex characteristics that are seen as biologically male versus in a testosterone low environment, the external sex characteristics that are seen as female. Given this is a highly gene-driven event, you can imagine that mutations in genes that are involved in sex determination will lead to atypical sex relative to genetic sex. And this set of rare disorders is called differences in sex development. Historically, it was called disorders of sex development, but the community is starting to move towards the use of differences of sex development. There are 3 types of differences of sex development. There's sex chromosome DSD. And what that means is Turner syndrome XO. So you have an XO, you have a streaked gonad, and so therefore your ovaries are immature. That's an example of a DSD. Other examples of DSD are 46XY DSD. So these are individuals that have normal karyotype. However, their external genitalia doesn't match their genetic sex as XY. So these individuals will either present as entirely female or ambiguous genitalia. The third type of differences in sex development is 46XX DSD. So you can imagine this is the opposite. So in 46XX DSD, these are individuals who have mutations in genes where although they have a XX karyotype, they have external characteristics or organs that are more consistent with testis and male fate. So this is what an ovary and a testis looks like if you're looking at it under a microscope. So on the left is the ovary and the right is a testis. This is at 7 weeks of development post-fertilization. So these are human ovaries and human testes. So that event of sex determination drives an extremely large morphological change in the bipotential gonad to form either the ovary or the testis. And so what you're looking at here in pink are the NR2F2 stromal cells that are coming in from the mesonephros into the bipotential gonad. What's not stained here in these gaps would be the FOXL2 granulosa cells and the germline cells, so they're forming large nests. In the testis, in blue this time, we have the NR2F2 stroma that emerged in from the mesonephros. And within these tubules that are lined by laminin, we have laminin in both these cases. This is where the Sertoli cells live, those SOX9 positive cells that are found in the testis and are driving the male germline cells to become prospermatogonia, versus the FOXL2 positive cells here, which are instructing the germ cells to become oocytes. So we've studied gonadal development using human fetal tissue for many years, and this is one of the papers that we published, creating quite an extensive 10x genomics data set using human fetal tissue, starting at around 6 weeks of development post-fertilization, up to around 20 weeks of development post-fertilization. And we've published a number of studies using these data sets, looking at the way lineages form both in the testis and the ovary to instruct our differentiation strategies. I just wanted to show one of the images from this paper where we were looking at the emergence of the germline at 6 to 7 weeks post-fertilization, utilizing computational approaches where we called the mesonephros the beginning of development, which is GATA2 positive, and these would be common progenitors to the ovary and the testis, and then tracking the formation of the granulosa cells, which are marked by LGR5 and TAC1, and the Sertoli cells, which are marked by SOX9. So in these computational ways that we were trying to evaluate how these lineages form in the embryo, we predicted that the common progenitors would give rise to bipotential progenitors that express NR2F1, PDGFR alpha, and LHX9, and that these LHX9 positive cells would ultimately give rise to either the sex-determined granulosa cells or the Sertoli cells. So again, this is computational based on mathematics, but not visual. And so where we feel that spatial transcriptomics has an extremely important role to play is to evaluate the spatial orientation of the emergence of these cell types in these very, very early organs to form. And I should say these organs are probably composed of less than 1,000 cells. And also they're very difficult to dissect because they're attached to the mesonephros. So again, 10x genomics and single cell approaches where we're looking at single cell suspensions of cells have reached the limit of what we can actually do to understand lineage specification, which is where spatial has become so powerful for us. I also want to point out that we're very interested in a window of development that basically is almost inaccessible using human material. And so therefore, over the last decade, we've formed a large collaboration with the Oregon National Primate Research Center to understand events in early primate development. So what you're looking at here is an image of a day 28 embryo, which is at Carnegie stage 12. The bipotential gonad, if it existed, would be down here. It doesn't exist yet. Day 28 is before. But I just want to give you an image of what this looks like. If you take a cross section through the embryo at this posterior region, this is what the embryo looks like. Some landmarks that you're very familiar with, of course, is the neural tube. These tubule structures here are that mesonephros, and what we're pointing to with these arrows is the epithelial lining that will ultimately give rise to the nascent gonad. And so this is a histology section standing for SOX17, which is marking the germ cells that are migrating in from their extraembryonic location into the embryo, into the dorsal mesentery. These are marked in blue, and they are migrating towards the genital ridge. SOX17 also marks the hemogenic endothelium, which is why you see these cells in the dorsal aorta as well. They are not germ cells. So we've spent a long time figuring out in non-human primates where the bipotential gonad exists. So that was a Carnegie stage 13 embryo. This is a Carnegie stage 16 embryo. And what you're looking at here is, again, that mesonephric structure. And these are XY animals as determined by karyotype. You can see the beginning of the budding of an organ off the mesonephros. And so this is the budding testis that's beginning to form. Looking at this using immunofluorescence strategies, what you can appreciate at this Carnegie stage 16 embryo is that there's actually slight differences in different samples. And that's because this is a dynamic window of embryo development where where sex determination is occurring. So one thing you can appreciate, here's the mesonephros in both of them. Here's the budding day 34 testis. They are beginning to create laminin basement membranes in both these cases. But what we noticed was the NR2F2 stroma is very bright in this sample, where emerging from the mesonephros, whereas the region in where the laminin is enriched in this particular sample, the NR2F2 is turned off. So what we hypothesized is that this sample at Carnegie stage 16 is likely undergone sex determination, and the mesoepithelial character of these cells has given way to an epithelial-like sex determination. And to address that, we looked at SOX9, which is a marker of sex determination, and we were able to identify that in the later sample where the NR2F2 positive signal has been repressed, what we can identify is the turning on of SOX19. Whereas in the other day 34 sample, again at Carnegie stage 16, what we can see is these regions of the gonad still have low levels of NR2F2. We can see the germ cells as marked by VASA. So this is a bipotential gonad that hasn't undergone sex determination at this stage. And this is a staining that was done by Enrique Sosa, who is here. So I hope I've convinced you that looking at these extremely small samples that are very, very rare to get would benefit tremendously from being able to understand some of the genes that are expressed during this window of time that can help us with our in vitro differentiation strategies, but also potentially help the field of differences in sex development, where the majority of cases are undiagnosed. And so what they do need is new biomarker discovery in order to have new candidate genes in the sequencing approaches of these individuals. So what we decided to use was the Visium CytAssist spatial transcriptomic technology. And so this is where I start hitting into lab meeting, because remember, I only have one set of data to show you, but I wanted to do the setup. We decided to use the Visium spatial transcriptomic technology because we were willing to sacrifice single cell resolution at this point in order to do as best as possible whole transcriptome discovery, because very little is known about any of the RNAs that are expressed in this window of time, because these samples are extremely difficult to get. So Visium, of course, this technology uses mouse and human. We don't have human, we have non-human primate. And so, so this is one of the caveats of the technology, I believe, so far, is that there's only 2 species. But biomedical research involves more than 2 species. And I think that there would be a benefit for these companies to start thinking about, rather than us building custom everything, is actually building platforms that more scientists can use. So our sample preparation is on formalin fixed paraffin embedded tissue. We, with all of our early embryonic samples, we're sectioning from tail to the heart. And so essentially we have 500 serial sections in a row where we're looking for certain organs in these serial sections. These sections are 5 microns in diameter. So we have a lot of section material to work with. We move through this Visium CytAssist workflow, the deparaffinization and imaging, the cross-linking and probe hybridization, and then transfer of the probes to the CytAssist slide, library construction, sequencing, and data visualization. So what I wanted to show you is just some analysis right off from our computational biologist on what the spots actually look like on the Visium platform on these rhesus tissues. And so these are 3 different samples: the bipotential, the determined, and then ultimately the testis. We identified across these whole sections around just under 1,000 to just under 2,000 spots. And of course, that doesn't represent the bipotential gonad, that represents the slice itself. One of the first questions we wanted to ask in these particular samples was about SRY expression, because it's known in the mouse and there's limited data in the human that SRY expression, which drives testis formation, is a gene that comes on rapidly and then is shut off rapidly, and SOX9 takes over as fortifying the testis program. So we were delighted to see, when we're looking at just the testis regions of this sample, which is next to the mesonephros, that in the bipotential gonad, there's low to no expression of SRY. As we move into the determined gonad, where we start to see SOX9, we can begin to see SRY expression within this region of the embryo. And then at the Carnegie stage 20, the day 41 testis, we can begin to see SRY turning off. So this dynamics is absolutely expected for the way sex determination occurs in an XY embryo. In order to evaluate the emergence of testis identity, what we wanted to do was to compare the mesonephros, which is where this organ originates from, to the developing testis itself, the bipotential, the determined, and the testis. And so we looked at differences in gene expression patterns between the mesonephros and the emerging testis to see what new genes and transcription factors to define this state in development. And so what we're able to identify is over these different data sets, between 267 and 483 differentially expressed genes in these three different samples. What we were really surprised by, but perhaps not in hindsight, is a lot of these differentially expressed genes turn out to be known differences in sex development genes. So what we believe is that this platform of spatial for looking at this stage in development will lead to the discovery of new genes that could be utilized to understand and help diagnose cases of differences in sex development, while also helping us to understand our differentiation strategies. So this is just showing some patterns of known genes and differences in sex development that are highly specific to the testis. And these are some of the candidates that show very similar types of expression patterns, including some like this one, MUT-S2, which is also found in the neural tube, and individuals that have differences in sex development. There are also some examples where they have neural tube defects as well. We also did the same experiment on the ovary and looked at the differences in expression between the mesonephros and the emerging ovary. And again, we also found a number of known differences in sex development genes that have been identified in patients with DSD. And again, showing you that no genes are very, very specific to the developing ovary and not expressed within other regions of the embryo. And we do have some examples of that in our candidates, and whereas others are slightly more widely expressed that we can now look at in our differentiation strategies. So with that, I'd like to end and summarize by saying engineering reproductive organs from stem cells requires knowledge of developmental biology, but I'd like to widen that by saying engineering any organ from stem cells, iPS cells, requires knowledge of how that organ forms in vivo. And we use spatial transcriptomics to understand how the ovary and the testis programs form from the mesonephros. Through this, we identified a number of DSD genes, and we think this is really significant because around 40% of cases have no genetic diagnosis, particularly a 46XY DSD. We think spatial transcriptomics at this time point could serve as a new strategy for increasing the diagnostic pool for these patients. And we do believe that at this next step, even though this was a review of how we view spatial transcriptomics, is to go down to single cell resolution to see how these different cell types are emerging. And with that, so this is my current group. Doing human and non-human primate work takes a village and also a relatively large amount of funding. So this is all the different funds that have supported our work with human and non-human primate over the years. And I'd in particular like to thank Sissy Wamaitha, who is also here. She took on the spatial transcriptomic part of the project about 6 months ago. So I've shown you every piece of data we have right now. And Enrique Sosa, who is a project scientist who's worked with me for a very long time, in particular on the primate projects as well, and he's also here. So with that, here are my collaborators, and I'd be willing to take any questions. So thank you. Thank you so much.
So it's a question about the determination aspect. So in zebrafish, the mechanism of determinations pretty much obscure. As far as I'm aware, the best guess right now is that it's the amount of primary germ cells that exist. I realize that in it's genetic determined, but is there any evidence that the number of male primary germ cells might have an influence on success of this development?
Yes.
Explain some of the non-genetic.
Yeah. Well, so zebrafish is not environmental either. It's and so different animals have different approaches for sex determination, but I'm not aware that it's to do with the number of germs. So, but I might be wrong.
Is there any evidence for number in that?
No. In fact, if germ cells don't, this is actually really interesting. So if germ cells don't make it to the ovary, or if they die, or ovary or testis, or if they die, then the somatic compartments disappear. And so what you're left with is gonadalogenesis. So germ cells being in this organ is absolutely critical for the somatic cells to coalesce around them. And there's a lot of crosstalk. But if germ cells aren't there, then you're left with strict gonads in both sexes. Yeah. Thank you for your beautiful talk.